So Ugur, yeah.
Okay. I would like to thank the organizers, particularly Christa Ma, for initiating and setting up this wonderful meeting. And I would like to introduce our genomics engineered personalized cancer and immunotherapy approach. So I hope this works. So if we look for the clinical care of cancer patients, we have at least two challenges. The first challenge is if we treat patients with advanced cancers, we have some patients having clinical benefit to the treatment, and some patients have showing a primary resistance to treatment.
Use the microphone, you're hard to hear.
Okay. Okay, I will try to come. Is it better? Okay. Okay, the first challenge for clinical care of cancer patients is that we have the situation that some cancer patients have a clinical benefit, but the majority of cancer patients often for solid cancers do not have a clinical benefit. And the second challenge is the recurrence of tumors in patients showing a clinical benefit. So we have a situation that we have to understand the individual variation of in the response of the patients, and of course what happens here during tumor progress. And if you look to the molecular picture, we have several levels of complexity. The first level of complexity is the germline variation, for example, the HLA antigens, the genetic polymorphisms. Of course, in cancer, we have the mutation heterogeneity. Cancer occurs is a mutation-based disease, and it is. There are two types of challenges. First of all, every cancer patient has a different set of mutations. And the second challenge is that every tumor cell is different from another tumor cell. So this is the tumor heterogeneity. But we have also a third level of complexity. This is the immune system. And the immune system cares also a clonal heterogeneity of T cells and B cells recognizing different antigens. And during the last years, we learned more and more that the immune system is able to recognize cancer and edit the molecular profile of cancer. And we also learned during the last decades there's an immunosuppression. And this axis of interaction, the immune oncology axis, is now becoming one of the most important treatment axis of cancer. We have the immunomodulators interfering with the immunosuppression of the tumor, and we have antigen-specific immunotherapy like adaptive cell immunotherapy being able to increase T cells and get effects on tumor cells. Of course, it is important to understand what happens at this axis. What are the molecules recognized by the immune system? And early work indicated that tumor mutations, cancer mutations, are recognized by T cells, and not only CD8 T cells, but also CD4 T cells. And further work, also from the Thomas Wolfes group, indicated that in some patients, this response, the immune response against mutated tumor antigens, might be the dominant ones. In the last 2 years, a number of studies indicated that the recognition of mutated tumor antigens may be prime for successful cancer immunotherapy. For example, Rosenberg's group showed that by exomic sequencing that patients with adaptive T-cell therapies recognizing mutations respond better to the treatment than patients recognizing non-mutated tumor antigens. A second study showed that patients responding to anti-CTLA-4 treatment have an increase of mutation-specific T cells. And a third study coming out 2 weeks ago showed that single expansion of antigen mutation-specific T cells can control patients with epithelial cancers. So the question is, if mutations are indeed, recognition of mutations are indeed key for cancer immunotherapy, Can we use that approach and induce de novo immune responses and amplify existing immune responses against mutations? And the concept arising here is the mutanome concept, starting with the sequencing of a patient's tumor, getting the mutations by comparing tumor sequence and the normal sequence, and then production of a vaccine, in our case of an RNA vaccine that allows to address multiple mutations. So this type of treatment would allow to induce an immune response specifically to the cancer mutations of the patients. We have started that approach in 2010 and were successful in showing the preclinical concept of the IVEC mutanome approach in a melanoma model where we sequenced the B16 melanoma, identified the mutations, generated peptide-based vaccines, and tested them for anti-tumoral prevention and therapeutic assays, and showed that about 20% of these mutations are recognized by T cells, and that these T cells are able to control cancer growth. And of course, the question is how to translate that approach into the clinical testing. We have implemented clinical grade, GMP grade or GCLP grade sequencing in Mainz, where we implemented sequencing, mutation analysis, prioritization of identified mutations by looking for MHC presentation and other factors, and we use Sanger sequencing for confirmation, getting some sort of a list of mutations which can act as a target list for developing of vaccines. This is the approach for SOP approach implemented in the labs for getting the sample and doing the sequencing, doing the confirmation, and going to the next steps of GMP production of the vaccines. So next, we looked for whether patients, and this is the case for melanoma mutations, we checked whether there is an pre-existing immune response in the peripheral blood against these mutations. And we were able to confirm in about 50% of the patients that against the predicted mutations, we have an existing immune response in the peripheral blood. So the peripheral blood is suitable for immunogenicity testing of mutations. And these mutations would be, of course, of particular interest because they are already showing an immune response. But I would like to emphasize that also de novo induction of immune responses might be feasible. As a vaccine platform, we use mRNA vaccines. So mRNA vaccines were introduced by Eli Gilboa's group using mRNA transfection into dendritic cells and using dendritic cells for adaptive transfer. And we modulated that approach, and we are using optimized mRNA vaccines that are targeted directly in vivo into dendritic cells to induce a strong immune response. For that, we are using mRNA, which is optimized in a sequence manner and in a chemical manner. There are a number of independent modifications leading to increased stability, increased expression, and increased immunogenicity of the mRNA, together resulting in an increased potency of such mRNA. And we developed formulations to bring such mRNA directly into dendritic cells, which are the key regulators of adaptive immunity. For that, we use intranodal vaccination, and the second, for the systemic applications, we have developed liposomal formulations that can be combined with the mRNA and bring directly mRNA into systemic dendritic cells, as shown here in mice models, where we intravenously inject such liposomal mRNA complexes, and the mRNA went to dendritic cells, mainly in spleen, bone marrow, and also in lymph nodes. And by depleting the dendritic cells, in that case, in a genetic model with diphtheria toxin, we lose complete the immune response. So this is a vaccine that not only enters dendritic cells, but because mRNA is able to activate TLR7 and TLR3, it is able to mature the dendritic cells. So we have the constellation of a vaccine encoding in an antigen and directly also maturating the dendritic cells, resulting in maturation signals and upregulation of type 1 interferon. And such type of vaccines are extremely potent. We see results, anti-tumor responses with these vaccines, which are not usual for vaccines. We see that with such type of potent vaccines, we are able to get rid of macroscopic tumors having sizes bigger than one cubic centimeter. That means combining the right antigens, in that case HPV antigen, with such a vaccine may be suitable for even controlling advanced tumors. Of course, we are testing this approach and combining this approach with mutated epitopes identified by our mutanome approach, and show that established metastasis here in a lung cancer metastasis model can be controlled by such a vaccine. So the question, of course, is how to put multiple epitopes into a vaccine, and our approach is the polytopic mRNA approach. That means we take several epitopes and combine them with non-immunogenic linkers, and these are combined into a vaccine cassette, and the vaccine cassette can be produced under GMP within a few days. So the production and release of such GMP vaccines can be done within 2 weeks. So the whole process, of course, is a novel approach, and together with the KIMT regulatory research group, we were able to work out a concept for fully individualized, actively individualized vaccines, and this concept was discussed with stakeholders from academia, industry, and regulators, including a meeting with the EMA in January 2012, and is now published as a framework for developing actively personalized cancer immunotherapies. Using that approach, we were able to convince the regulators to approve the first clinical trial in melanoma of a mutanome vaccine. This is the approach. Get the mutations and use the mutations to make polytopic epitopes. We have the allowance to get up to 10 epitopes for vaccination for our first clinical trial, and the approach for sequencing and producing the vaccine takes about 3 months. Within this 3 months, we are vaccinating the patients with non-mutated shared tumor antigens. For melanoma, we use tyrosinase and New York ESO, and after the vaccine is available, we start vaccination with the mutanome vaccine. So it's a running study. We have our first 5 patients in the clinical trial. The first patients have got the mutanome vaccine, and of course we are curious to see the results. So in collaboration with our partners from Heidelberg, a similar approach is done for glioblastoma. I assume that Hans-Georg Rammensee will tell something about that approach, and we are using that approach also for triple-negative breast cancer. We have an EC-funded consortium addressing that. And the question is, we are here in a colon cancer meeting, is this applicable also for colorectal cancer? We believe yes. Colorectal cancer, if you look to the number of mutations, is a cancer type having having many patients with high numbers of mutations. We have tested colon cancer model. CT26 is a well-known model. Got the mutation map for CT26 and identified the shared tumor antigen map for this tumor too, showing us that many shared tumor antigens are expressed in this tumor type, and we have performed vaccination with shared tumor antigens, showing that the mRNA vaccine approach is able to control this tumor, and we have performed also the mutanome polytope approach in established lung metastasis, resulting in complete regression of lung metastasis in this model using purely mutated epitopes. So this approach might go into clinical testing in colorectal cancer too. So what are the conceptual advantages? The conceptual advantage is that currently we have the situation that every patient has another marker set. And usually for stratified medicine, development is focusing on the intersection of epitopes. With the mutanome approach, we can really take advantage of every individual set of vaccines. So we have much more opportunities by genomics tailored individualized treatments. And having this many opportunities might allow us to get a key advantage. This is the deep sequencing study, or ultra deep sequencing study of a tumor, showing you that a tumor has, of course, dominant clones, but it has many subdominant clones. If you focus your treatment only on this dominant clone, then of course you may get a clinical response in getting rid of the dominant clone, but these clones may go up. And the promise of such mutanome vaccines are that we are working with polyspecific immunotherapies, and we may be able to control all clones of the tumor. We will see whether this is true. The clinical studies are ongoing. Thank you for your attention.
For this really impressive talk. Having heard the previous speaker, Nils Hallamar, seeing all the T cells at the invasive margin, do you think it's good enough to activate, stimulate the cells that are sitting there? Would they then infiltrate the tumor and get rid of the tumor, or do you need like a combined treatment approach when you think of your vaccine idea?
This is an excellent question. I believe that the T cells are there and doing nothing does not exclude that by stimulation in the lymphatic system, you can really reactivate the T cells. So this is one of the key questions. The T cells in the tumor are just directly in contact with the antigen. Having the antigen in the lymphatic system, and this is an important message coming from many mice experiments, might have a difference. And of course, if this do not work alone, combination is possible.
Other questions? You mentioned for such an individualized strategy, doing exome sequencing, looking at the most immunogenic mutations, then designing an indeed patient individual vaccine cocktail. You only need 3 months. Is that realistic?
Yeah, it is. At the moment, it's 3 months. Of course, we would like to reduce this to less than 1 month, but it should be possible. At the end of the day, the time for sequencing will be more rapid in future, and production of mRNA is not limiting.
How is the mRNA stabilized and then internalized?
Okay, the mRNA is not stabilized for degradation by RNases. It is stabilized pharmacokinetically after entering into the cell. We have included at the 5' end of the mRNA a chemical modification which counteracts the 5' degradation of the mRNA. We have included UTR optimizations allowing us to get very long half-life of the mRNA, and we are protecting the mRNA by liposomal formulations that protect the mRNA from degradation by plasma ribonucleases.
Just one quick short question. You're focusing on your sequencing on non-synonymous SNPs inside the exons, presumably because you're looking for immunogenicity changes. What about SNPs in the introns that actually change the splicing pattern that still change the coding sequence?
Yes.
These always seem to get ignored.
It's an excellent question. So we started in focusing on the SNPs in the. We are now working on indels. We are looking, of course, in mutations in other regions, and we are combining that with mRNA sequencing to get really the full picture of potential alterations.
If you look at 20 patients with advanced diseases, what tumors whatsoever, and you have the exome sequencing, you have your list of the most potential immunogenic mutations, and then you test peripheral blood. In what percentage of patients do you find really a mutation-specific detectable T-cell response?
At the moment, we have about 50%.
Really? In the periphery?
Yeah, but we are testing about 50 mutations per patient, and 50% show a mutations against one or more in the set of the mutations.
I don't see any other questions, so thanks again.
Thank you.